One day closer to the exam. One day closer to showing me off how much you've learned. That's good, right? Nobody likes that one. I have decided that we will finish the material for this exam with where I finish here talking about non-competitive inhibition. So very early in today's lecture, that'll stop. And I'll mark that on the highlights as well. So you'll see in the highlights where the material stops. I have also scheduled a review session, and that will occur tomorrow evening at 7 p.m. in ALS 4001. Okay? So ALS 4001. I will videotape that, and barring any technical difficulties, get that posted as quickly as I can. Usually that would be posted by certainly, what is tomorrow is Wednesday, sometime on Thursday. I might even get it posted by sometime tomorrow night. We'll see. Okay? All right, any questions? Concerns? Thoughts? Anything? No? Okay. Well, let's finish up talking about the inhibition of enzymes. And yesterday I talked about competitive. And so just to remind you a little bit about these different types of inhibitions, let me just step you through it as I promised I would do. So competitive inhibition occurs when a molecule resembles a substrate and competes with the substrate for the binding site on an enzyme. Okay? So competitive inhibition, the inhibitor will resemble the substrate, and it will compete with the substrate for binding to the active site on the enzyme. All right? They both are trying to get to the same place. And so as I talked about yesterday, if you have a lot of substrate, then the substrate is going to win that competition. The substrate is going to bind. If I have 50 million molecules of substrate and I have 500 molecules of inhibitor, the likelihood that the substrate is going to get there is very, very high. The likelihood that the inhibitor is going to get there is very, very low. Okay? As a consequence of that, we see that the maximum velocity for a competitively inhibited reaction does not change, because it means that we can effectively outcompete the inhibitor. If we add enough substrate, we will outcompete the inhibitor, and the inhibitor will therefore not be able to do its thing. Okay? That's not the case with non-competitive inhibition. That is not the case with non-competitive inhibition. With non-competitive inhibition, what we see is that there's no competition. It doesn't matter how much substrate I add, I'm always going to have a fixed percentage of the enzyme that is going to be inhibited. That percentage was changing when I had a competitive inhibitor, because the more substrate I added, the less the effect the inhibitor had. Right? So for non-competitive inhibition, I've got a fixed percentage of enzyme that's going to be inhibited. Doesn't matter how much substrate I add. Does that make sense? So let's say I have 50,000 molecules of enzyme, and I add 20,000 molecules of inhibitor. Okay? Effectively, that means that every time, every reaction I do, 20,000 molecules of enzyme are going to be inhibited. Okay? I'm only going to have 30,000 molecules of enzyme in every reaction. Right? Doesn't matter how much substrate I add, every reaction is going to have only 30,000 molecules of enzyme. All right? Everybody buy that? What happens if I reduce the amount of enzyme that I'm using in a reaction? What happens to Vmax? Does it stay the same? Or does it go down? It goes down because Vmax is a function of how much enzyme I've got. Remember that? So that's what a non-competitive inhibitor is doing. It's effectively reducing the amount of enzyme I have in a reaction. Therefore, Vmax for a non-competitive inhibitor decreases. Vmax for a non-competitive inhibitor decreases. That's not the case for a competitive inhibitor. Competitive inhibitor, Vmax stays the same. Okay? Now, the last point I'm going to tell you, I will explain to you later if you'd like to understand, but a lot of students just decide they want to memorize it, and that's fine too, because it's a little bit hard to understand. Non-competitive inhibition, the Km does not change. The Km does not change for non-competitive inhibition. Now, as I said, I'm not going to explain that here. It's not complicated, but it's more time than I want to take here, and it's not a major point. Okay? And not a major point in terms of explaining how. So we've seen two types of inhibition. I'm going to summarize them here. Competitive inhibition, we saw no change in Vmax, but we saw an increase in Km. That's what happened with a non-competitive, with a competitive inhibitor, right? With a non-competitive inhibitor, we saw exactly the opposite. We saw Vmax decrease, but we saw Km did not change. Okay? Very different effects these two types of inhibitors have. Okay? Questions about that? None? Yes?
Sorry, I was thinking out.
You're just like me.
Well, I was so for competitive, Vmax doesn't change, but the Km increases?
Yes.
Okay.
So for competitive, Vmax does not change, but the Km increases. For non-competitive, the Vmax decreases, but the Km stays the same. Okay? All right, folks, that's the end of the material for the first exam. Woo! Celebrate, right? Okay. Excellent. What's that? That's actually what the Lineweaver-Burke plot looks like. Maybe I should explain that Lineweaver-Burke plot to you. Okay? Students get confused about a Lineweaver-Burke plot for non-competitive. Here's the uninhibited reaction right here. Here's the competitively inhibited. You'll notice they both have the same Km over here. They're the same minus 1 over Km, which means they have the same Km. Why is it the top line instead of the bottom line? Why is the non-competitive inhibition the top line? Anybody know? What's the y-intercept? What's the value of the y-intercept? It's 1 over Vmax, and it's because it's the reciprocal. That's right. So you take the reciprocal of a small number, you get a larger number. You take the reciprocal of a large number, you get a smaller number. So Vmax goes down, but 1 over a decreasing number means an increasing value on the y-axis. Okay, so that's the end of the material for Exam 1. Okay, well, I told you that the Exam 1 was pretty calculation, blah, blah with that. So hopefully you won't get too snowed by the calculations. If you're having difficulty understanding the calculations, come see me. I've had a couple of you already do that. I'm more than happy to work with you and to help you to better understand how the calculations are done. All right. So for the new material here, I want to talk about the control of enzyme action. And the control of enzyme action is very important, just like controlling that Maserati going to Winco is important. If we don't control it with a speed limit, we're going to probably run over some things on the way. And if we don't control enzymes, we're probably going to break or make something that we don't want to break or make, right? Because they work so fast, we've got to be able to have some controls on them so they don't go overboard. If they start going overboard, we're going to have some troubles. And we're going to see that cells have some very, very good controls and some very clever controls on enzyme action. Okay? So we're going to spend a little bit of time talking about those. One of those I've already talked about. Let me show you something that'll just kind of go, wow. All right? I talked about ATCase the other day. I said that it was an example of an allosteric enzyme. And ATCase catalyzes this reaction that you can see going over here. I don't care if you know that reaction. It doesn't matter. Okay? I do care that you know something about that reaction. All right? So let me give you some background. ATCase catalyzes the first reaction in the synthesis of CTP. CTP is a nucleotide. It catalyzes the first reaction in the synthesis of CTP. Now, you'll notice I said the first reaction. When we make nucleotides, we start with very simple things, and it takes a series of reactions. It actually takes 10 reactions to get the final product, which in this case is CTP. There's an extra C in there, isn't there? Cytidine triphosphatate, phosphate, what is, I guess, whatever that says. All right. And anyway, it takes a series of reactions, 10 reactions, to get here. Why do I tell you that? Well, it turns out that ATCase is a really interesting enzyme. There are several things that can bind to it that affect CTP's, it affects ATCase's activity. Okay? One of these is the end product of the pathway. I'm going to repeat that. One of the things that can bind to ATCase and affect it is the end product of the pathway. The end product of the pathway is CTP. Now, I'm going to show you in a minute why that's significant. All right? But for the moment, you can just say, okay, ATCase binds to CTP, and when it binds to CTP, the enzyme is inhibited. Okay? ATCase binds to CTP, and when it binds to CTP, the enzyme is inhibited. Now, this turns out to be, it's a very simple fact. You can sit, you can memorize that fact, but the really cool thing about that fact is the fact that CTP is the end product of the pathway. Every enzyme, or every reaction in this series of reactions, is catalyzed by a specific enzyme, meaning that there are 10 enzymatic reactions that occur in going from here at the top down here to CTP at the bottom. If the cell were a total control freak, and by the way, cells do tend to be control freaks, okay? If the cell were a total control freak, what would happen is the cell would have a control on every single enzyme in that pathway. And if you think about it, that gets kind of complicated. I've got to control enzyme number 1, I've got to control enzyme number 2, I've got to control enzyme number 3, et cetera, and I have to control the last enzyme as well. What if I just controlled the first one? If I just controlled the first enzyme and the first enzyme got stopped, wouldn't that mean that there wouldn't be any product so that the next reaction wouldn't have anything to work with and the next enzyme wouldn't have anything to work with? By shutting off the spigot, I shut off the lawn from being watered, right? I'm turning off the spigot, and the spigot is right here. The cell can control one enzyme and control an entire pathway. That's a very powerful concept, a very, very powerful thing. It makes for simplicity, but it also allows for some pretty cool stuff. Why is this important? Well, if we think about CTP building up, if the cell is making too much CTP, there might be consequences. And it turns out that there are two consequences if the cell doesn't control this pathway. One of the consequences is, whenever you have an imbalance of nucleotides, you make mutation much more likely. So if I get too much CTP, the cell is going to become a mutant zombie from hell. Okay? We don't want cells to be mutant zombies from hell because they become known as tumors. Right? The other reason that we don't want this pathway going out of control is this pathway requires a lot of energy. The cell is going to waste energy if it doesn't shut this pathway off when it's got plenty of CTP. Well, the beauty here is that this pathway shuts itself off. When the CTP concentration gets too high, it binds to the first enzyme in the pathway and shuts down the whole pathway. There's beautiful balance here. This phenomenon I've just described to you has a name. It's called feedback inhibition. Feedback inhibition. What's happening is the end product of the pathway is feeding back and turning off the first enzyme in the same pathway. Perfect balance. Too much CTP, pathway turned off. Too little CTP, pathway turned on. Now this enzyme, I said, was a really interesting enzyme, and this is one of 3 reasons that this is an interesting enzyme. There are 3 things that control this enzyme. CTP is one of them. The second one is another nucleotide. Okay? Another nucleotide can bind to this enzyme and affect this enzyme's activity. The other nucleotide is ATP. Right? Now we're going to figure out in a minute if it turns the enzyme on or it turns the enzyme off. Let's think about DNA and RNA. Everybody learns in high school, that A pairs with T and G pairs with C, correct? Does everybody remember how the nucleotides are categorized in terms of purines and pyrimidines? Purines are what? A and G, okay? And the pyrimidines are C and U and T, right? So, CTP is a pyrimidine nucleotide. Pyrimidine nucleotides in nucleic acids are complementary to purine nucleotides. That's why T pairs with A, or U pairs with A, and C pairs with G, right? Okay? If we have too little pyrimidine, that means we have too much purine. Because the comparison of the two, if we have too little of something, we don't have enough, we have too much of the other. All right? If we have too much purine and not enough pyrimidine, we would want to turn on pyrimidine synthesis, correct? So you tell me, what does ATP do? It turns on this enzyme. ATP turns on this enzyme. Now, ATP is a purine. It's used to make nucleic acids. But ATP, we also remember, is important because it's a measure of the cell's energy. The more energy a cell has, the more ATP it will have. Now, energy turns out to be really, really important for cells. The reason it's important for cells is because cells need it to do all the things that they're going to do, including division. Cell division, if you think about it, is a very energy-requiring process. You have to completely duplicate the chromosomes. You have to go through a pretty elaborate process in order for this to happen, and that's going to require a lot of energy. It's also going to require a lot of nucleotides. When cells have a lot of ATP, look what they're doing. They're turning on the synthesis of pyrimidines. They're getting ready to divide, folks. Pretty cool. Okay? We see, in fact, we'll talk later in the class about nucleotide metabolism, but we see great balance of nucleotides because nucleotides are intimately tied to every cellular process. ATP is a very common energetic one, but we'll see GTP is also involved in energy. We'll see CTP is involved in the synthesis of glycerophospholipids. And we'll see, you don't need to memorize this, and you'll see UTP is involved in the synthesis of glycogen. All of the nucleotides have some cellular role besides being in RNA, for example. Okay. Any questions about what I've just been talking about here? Yes? ATP turns on ATCase. So ATP is a purine. And so if I have a lot of purines but not enough pyrimidines, I want to make more pyrimidines to balance, right? So that's why turning that on makes good sense. On the other hand, if I have too many pyrimidines, they turn themselves off. You look puzzled. It needs to sit there. Okay. Well, think about that. If you have questions, let me know. How's that? Other questions on that? Okay. Now, I said there are three things that, here's a good representation. This shows the entire pathway. There's actually, I think it's 10. It depends on where you start counting. But there are several reactions here that are important for the cell to control because they're going to waste energy if they're running reactions they don't want to run. They're going to favor mutation if they make too many pyrimidine nucleotides. Now, the third thing that affects this enzyme is actually the substrate, aspartate. All right? So you see that aspartate is one of the substrates. And by the way, remember now that CTP is not the product of the enzymatic reaction. It's the product of the pathway. Okay? The product of the enzymatic reaction is this mouthful guy over here. A common mistake I see students make on their exams is they want to tell me that CTP is something that the enzyme makes, and it doesn't. Okay? The pathway makes CTP. All right. But anyway, this guy right here, aspartic acid, is also something that affects the enzyme's activity. It turns out that aspartic acid turns the enzyme on. Aspartic acid turns the enzyme on. What do we use aspartic acid for? What do we make with aspartic acid? What's that? I'm sorry, I didn't hear you. Artificial sugar, well, actually, that's true. But aspartic acid and phenylalanine. But in terms of our cells, what are our cells making with aspartic acid? Proteins, right? It's one of the amino acids. A cell has got to divide. A cell wants to have enough energy before it starts the division process. Do you suppose a cell would want to have enough amino acids before the division process? Absolutely. And how does it measure those? With one of the amino acids right here. If the cell has plenty of amino acids, one of the signs will be that it has a good amount of aspartate. And aspartate is telling the enzyme, hey guys, let's make some pyrimidine nucleotides because we're going to have fun. We're going to go divide. Happy hour. Right? Make sense? Okay. All right. So aspartate turns ATCase on. CTP turns it off. ATP turns it on. Okay. Three things. Really remarkable enzyme. I showed you earlier that ATCase has a sigmoidal plot, and we talked about the significance of that sigmoidal plot. The significance was that this meant that, first of all, that there was binding of one substrate was affecting another, just like hemoglobin. And that meant that we had to have multiple subunits. All right? ATCase is pretty remarkable. It has 12 subunits. 12 subunits. 6 of the subunits catalyze the reaction. The other 6 subunits bind to either CTP or ATP. So we break the subunits into two categories. Catalytic subunits, that's the first 6 that catalyze the reactions, and regulatory subunits, which are the ones that control the enzyme. ATP and CTP bind to the regulatory subunits. Aspartate binds to the catalytic subunit. Now, when I talk about turning an enzyme on and turning an enzyme off, we have a parallel another parallel that we had with hemoglobin. With hemoglobin, we talked about two different states. What were the two states of hemoglobin in terms of structure? R and T. We see the same thing with enzymes. Enzymes that are more active are in the R state, and enzymes that are less active are in the T state. Binding of ATP or aspartate will put ATCase into the R state. Binding of CTP by ATCase will put it into the T state. Yes? Say it one more time. Yeah, a lot of names and everything. I'll stop here in a second. Okay. So, binding of ATP or aspartate puts the enzyme into the R state. Binding of CTP puts the enzyme into the T state. You guys look like you're needing a joke. A joke? Okay, let's see. Okay. This is a joke that a student told me, and I kind of like this joke. It's kind of cute, okay? It's the crunch bird joke. How many of you have heard the crunch bird joke? Be honest. Okay. So there's this lady, right? And this lady decides that it's her husband's birthday, and she wants to go get him something cool and exotic for his birthday. And so she goes down to the pet store, and she tells the pet store owner, she says, I want to buy something for my husband's birthday, but I don't want the usual gift. I want something different. And he says, piece of cake. I got it for you. He goes back, and he comes out, and he walks out on a leash. He has this iguana, about 6 feet long, giant lizard-like thing, right? And she looks at it and says, oh, I don't think I want that around the house, you know. He says, okay, no problem. He takes the iguana back. He comes out a little bit later, and he's wrestling this giant boa constrictor. He puts it down. Everybody's going, oh. And she says, I don't think I want that around the house either, you know. Okay, ma'am. So he goes back, and he begins to think he's a shoe salesman here, right? And so he picks up another animal and brings it out. No, same story. No, same story. This goes on and on and on. He's just about running out of animals. And he says, I know what you want. Okay? You want a crunch bird. And she says, what's a crunch bird? And he says, look over there in the corner. She looks over in the corner, and there's this little tiny bird sitting on a perch. All right? And this little bird is sitting there. He says, watch this. He says, crunch bird. He says, chair. And the bird jumps up off of this perch and flies over to this wooden chair and demolishes the chair in front of her eyes. It's sawdust in just a very short period of time. She says, that's incredible. He says, that's nothing. Crunch bird, desk. And there's this great big wooden desk, and this bird flies over, and the same thing. And she's going, that's absolutely incredible. She says, I've got to have that. It's the exotic thing that she wants for her husband. So she grabs the bird, pays for the bird, and takes it home. And she walks in and says, honey, I got something for your birthday. He goes, oh. He said, what is it? She said, it's a crunch bird. He looks at this little thing on her finger and he says, crunch bird my ass. You get the idea. Okay. Okay. Everybody awake now? There's the exciting structure of ATCase. At this point, who cares, right? You've got 6 regulatory subunits, you've got 6 catalytic subunits, and that's what I've already told you. Okay. Let's think about this. Tell you what, let's not think about that, all right? Try to think of a way that I can make this One of the things I find is when I teach this part of the thing, students frequently go, oh man, it's so hard to understand. I'm trying to think of a different way that I can make you understand it. All right, so let's try a little experiment here. We talked about how hemoglobin bound one oxygen, and that favored the binding of a second oxygen, and a third, and a fourth, right? And we call that phenomenon cooperativity, right? The binding of one favored the binding of others, right? Okay? When we think about an allosterically controlled enzyme, and we've seen a lot of parallels to hemoglobin, we can think of a same sort of thing. The binding of one may favor the binding of another, et cetera, et cetera. But we also now have to account for inhibition as well. Okay? The binding of one might inhibit further binding, et cetera, et cetera. And in fact, that does happen. Because what's happening is the binding of an inhibitor converts the enzyme into the T state, and the T state was just like hemoglobin. It doesn't, in the T state, hemoglobin didn't bind oxygen very well. In the enzyme, the T state doesn't bind substrate very well. Okay? The question then is, how do we explain this process happening? And there's really two ways of explaining it. One is what I will call a cause and effect way, and the other is magic happens. I'm trying something different here. Okay? Cause and effect versus magic happening. Cause and effect is really easy to understand. Cause and effect says I've got an enzyme, it's got multiple subunits. Binding of one favors the binding on the second, favors the binding on the third, et cetera, et cetera. And so the first subunit causes the second subunit to change structure so that it more favorably binds a second one, which causes the third subunit to change structure so that it more favorably binds something. And that's the cause and effect. The first one causes everything else to happen. That make sense? Cause and effect is fairly easy to see. It's fairly easy to conceptualize. Okay, so that's what happens with cause and effect. And that's what we call the sequential model of catalysis. It's cause and effect. It's sequential. Binding of the first favors the second, favors the third, favors the fourth, favors the fifth, favors the sixth, blah, blah, blah. Everybody with me? That's one way of explaining how these processes occur. Now, the magic happens, if I call it something magic, what does that conjure up in your mind? I don't know. I'm just sitting here and all of a sudden there was this rabbit in this box, right? Poof, there's a rabbit. That's magic. We don't ask how the rabbit got in the box because we say it's magic, right? Everybody with me? Okay. The other model of catalysis is called the concerted model. This is the magic model. The magic model says that enzymes flip all by themselves from R to T or from T to R. It doesn't take a cause and effect. In the case of the first thing I told you, the binding of one favored the binding of another, or the binding of one might inhibit the binding of another. We're going from T to R, or we're going from R to T. We can go either direction, depending upon whether we're binding an inhibitor or an activator, whether we're binding ATP or CTP, right? The magic model says, I don't know how it happens, but the enzyme flips from R to T on its own, and then when it binds the appropriate molecule, it gets locked in that state. So let's imagine I've got ATCase. It flips from the R to the T state. If, when it's in the T state, it binds to CTP, it gets locked in the T state. I might have ATCase that flips from the T to the R, and if it binds to ATP, it gets locked in the R state. With me? The magic is in the flipping. T to R, We don't ask how it happens, or R to T. We don't care how it happens in the concerted model. In the sequential model, we say R to T or T to R is caused by something, right? Cause and effect. The cause is the binding of something. If I bind ATP, it causes it to go into the R state. If I bind CTP, it causes it to go into the T state. The magic happens says R and T happen on their own. ATP locks it in the R state. CTP locks it in the T state. Make sense? If you guys buy that, that's really good. Any questions about that? Yes?
So ATP locks in the R state and CTP.
Locks it in the T state. That's right. ATP locks it in the R state. CTP locks it in the T state. So that's two ways of explaining activation and inactivation of an enzyme. Okay. That's all the blah, blah, blah that you see on the screen. All right. Well, allosteric control. Oh, yes, question? Two different ways of explaining a phenomenon. And it turns out that enzymes tend not to have only one. They actually have a sort of a blend of them. But those two different ways that I've described to you are two different ways of explaining activation and inactivation of an enzyme. Okay? Good. Allosteric mechanisms, and by the way, when I say allosteric, I'm going to give you a definition here. If there's one definition I ask on an exam more than almost any other definition, it's this one right here. Okay? What does it mean to say an enzyme is allosteric? An allosteric enzyme has its activity affected by the binding of a small molecule. An allosteric enzyme is an enzyme that has its activity affected by the binding of a small molecule. Depending on the molecule, that might activate it, it might inactivate it. But an allosteric enzyme is an enzyme whose activity is affected by the binding of a small molecule. Everybody with me? It's a good definition to know. Well, allosteric control is only one way of controlling enzymes. I'm going to talk about some others. Okay? Allosteric activity is only one way of controlling an enzyme. All right? Another way to control enzymes is to covalently modify them. That is to cause some chemistry to happen on the enzyme. Now, there are several ways of doing this. Several ways of doing this. I'm going to talk about two. All right? The most common of these is the first one you see listed, phosphorylation. The name tells you what it does. Phospho part telling you that there's a phosphate involved. The ylation part means it's getting put on. So phosphorylation involves putting on of phosphates. The reverse of that involves dephosphorylation, which is the taking off of phosphates. Now we're going to see what effect that has in just a minute. Okay? First of all, what does phosphorylation mean? Okay? I said it means putting a phosphate on. It turns out that there are 3 amino acids that commonly get phosphorylated, meaning they gain a phosphate. One is the side chain of serine. Here's a serine residue. Serine has a hydroxyl group. In fact, all 3 that get commonly phosphorylated have hydroxyls. The OH gets a phosphate put onto it. Look at the charge on that phosphate. Serine didn't have a charge. The side chain didn't have a charge. Now, when we put a phosphate on it, it's got 1, 2 negative charges there. We've changed the charge of a part of a protein. What happens when we change charge in a protein? We may change the shape. And if we change the shape, we may change the way in which the protein functions. Phosphorylation for some proteins turns them on. Phosphorylation for other proteins turns them off. It depends on the protein. We'll talk about different examples. You should know the name of the enzymes that catalyze phosphorylation. The name we give to enzymes that catalyze addition of a phosphate is kinases. K-I-N-A-S-E-S. Kinases. You're going to hear a lot more about kinases in the class as we go along. So this reaction that you see on the screen would be catalyzed by a kinase. And this reaction is very common. You'll see that here's phosphate on ATP. And ATP is donating not only a phosphate, but also energy for this reaction to occur. Okay? Here's another amino acid that gains a phosphate, and it's threonine. Threonine also has a hydroxyl group. And you can see a kinase does that, and we see the same result. The oxygen gets a phosphate put onto it. We're going to change the charge. And when we change the charge, we are going to likely change the way the protein acts. Serine and threonine are two. The third amino acid that gains a hydroxyl is tyrosine. Serine and threonine are chemically similar. Tyrosine is quite a bit different. Okay? But again, we see the hydroxyl gets the phosphate. Same thing as we saw before. Change the charge, may change the structure and function of that protein. Kinases catalyze all of those reactions. Okay? Well, I said earlier that cells are control freaks. Control freaks want to control everything about something, right? So if cells have a way of putting phosphates onto something, they also want to have a way of taking phosphates off of something. In fact, they need to. Let's say that we put phosphate onto an enzyme and it activated that enzyme. And remember, we're trying to control enzymes here. We said there may be times the enzyme will be active. We don't want it to be active. If we're going to have a way to activate it, we want to have a way to inactivate it as well. There's another group of enzymes that remove phosphates from things. Those enzymes have their own name. They're called phosphatases. P-H-O-S-P-H-A-T-A-S-E-S. Phosphatases. So a phosphatase would remove the phosphate off of this tyrosine and leave a hydroxyl. In other words, just making the reaction go backwards. Everybody with me? Now, one of the things that we will see before too long is that there's a process that cells have for talking to each other. Cells actually talk to each other, and they communicate information to each other. And one of the ways that they do that is by putting phosphates on or taking phosphates off of certain proteins. Putting phosphates on or taking phosphates off of certain proteins. That process is called signaling. And signaling is really just cells talking to each other. It's really all it is. Okay? Okay. Well, let's see an example of an enzyme that is affected by addition of a phosphate. Now, this looks pretty complicated, and I'm going to simplify it for you. Okay? What you see on the screen is an enzyme known as glycogen phosphorylase. Glycogen phosphorylase. It catalyzes the breakdown of glycogen. Okay? This enzyme is regulated allosterically and by covalent modification. It has both regulatory mechanisms that affect it. I'll repeat that. It's regulated allosterically and by covalent modification. And by the way, I should say that putting phosphate on is a covalent modification, and so is taking phosphate off. Anytime I'm affecting a chemical bond, it's a covalent modification. So putting a phosphate on and taking it off are both covalent modifications. This enzyme is pretty cool. It breaks down glycogen really fast, and glycogen is a polymer of glucose. We have a lot of glycogen in our liver. We have a lot of glycogen in our muscle cells. When you break down glycogen, you get glucose, and glucose is what we use to make energy for our muscles. So having glycogen in our muscles is important, and being able to activate that enzyme that breaks it down quickly is important. We've all heard stories about the parent that sees the child in danger underneath the car, and their terror level goes up, and they grab the car and they lift the car up. You've heard these stories, right? We've said they've had adrenaline. The stories are true. They can do that. Okay? They can do that. And they can do that because what's happened is adrenaline has another name, and it's a hormone we call epinephrine. Epinephrine is a way of signaling. Comes from our adrenal glands. Okay? It's released into our bloodstream. It goes to our liver cells. It goes to our muscle cells. And it favors the phosphorylation of glycogen phosphorylase. When glycogen phosphorylase gets phosphate put onto it, it becomes really active. It starts breaking down glycogen. And that's what you see going on here. Active, active. Okay? This is something else I'm not going to talk about, the T-state form of this. Okay? In summary here, to keep it simple, putting a phosphate onto glycogen phosphorylase makes it active. That's pretty cool. Okay? And it's happening because the bloodstream has gotten epinephrine, and epinephrine has told glycogen phosphorylase to break down glycogen. And by the way, we're in a lot of danger, so break down a lot of glycogen, make a lot of glucose, and get those muscle cells doing what muscle cells can do. Okay? Make sense? All right. I keep running over, so today I'm not going to run over. I will stop there unless there are questions. I have a song for you today. It's another one I can't sing. Most of them I like to sing, but this is another one I can't sing. It is... it's called The Way They Work. It's a hard one to sing. All right, see you guys tomorrow.